News is a graduate student organization. Our main goals are twofold. First is to bring awesome events like this to general public, where we bring scientists and training scientists to talk about their work with people like you. And second is to actually train the next generation of scientists to be better communicators. We do this through our seminar series, which you're at right now. And we also do this through an event called Science by the Pint. This is a little bit of a more informal event. It's basically we bring scientists in their labs to a bar, either the Burren or Aeronaut. We alternate each one every month. And they just come, share a beer with you, talk about their work. So if you're 21 plus and interested in that kind of event, please join us. The next one is Monday, June 13th at the Burren. And this one is entitled Fires, Invasives, and Ticks, Oh My, Forecasting Ecology in a Changing World. So it should be pretty interesting. And even though we're winding down with our spring lecture series, we have another very exciting event coming up. This is called DayCon, which stands for our Day-Long Science Conference, and is our second annual one. It's occurring on Saturday, June 11th. It's basically an entire day that's completely free with lectures, seminars, poster sessions where you can talk with scientists about their work. And this year's theme is Bodies in Motion, so it's all about athleticism, healthy aging, physics of sports, things of that nature. And I have to say, I've seen some of the practice talks so far, and they're very excellent. So if you're able to join us for some of that day or all of the day, please feel free to check out our website, SITNboston.com, where you can find more information about all these events. And we also have a blog that a lot of our speakers have written for that's really had a lot of exciting new topics on it as well. So without further ado, I'd like to introduce Nick. He's in the program in neuroscience, and he's going to talk to us about his work in studying instinct. And also, just so you keep in mind, there's going to be a couple of question breaks throughout. So please save your questions for those question breaks, and then Nick will be happy to answer any you have to ask. Great. Thanks, Nick.
Thank you. Hi, everyone. Thank you for coming. So today I'm basically going to talk to you about some of the things we've learned recently and some of the tools that have been developed and some of the things that are on the horizon in the study of the neurobiological basis of behavior. And so the talk is going to be broken into three general sections. So in the first part, I want to go over some of the basic concepts that scientists use to help them think about behavior when they're studying its biological basis. And so I'll give you some terminology and some concepts that help us think about behavior when we study it. And in the second part, I'm going to talk to you about some of the modern tools that have been developed that allow us to actually study behavior much more rigorously and much more systematically than we ever could before. And this includes techniques for not only watching animals behave, but also techniques for recording neurons while animals are behaving and actually manipulating circuits within the brain during ongoing behavior. So we can actually figure out what is cause and what is effect with respect to the circuits that are allowing animals to behave. And in the final part of the talk, I want to talk about some of the recent discoveries that have come from applying some of these tools to the behavior of awake animals. And I want to end with some of the implications that these tools and these results have or might have in human health. So part of the overall motivation for Science in the News and for this talk is to make you better equipped to understand some of the things that you're likely to read about in the headlines. And so a lot of the work that I'm going to share with you today, both in terms of the technical aspects that go into doing it and the actual results that I'll share with you, is stuff that often gets coverage in the press. And so you see a lot of really flashy and provocative titles about mind control or switches in the brain that turn something on or off, and these weird images of rats and things with glowing objects on the top of their brains. And so what I'm going to do is actually kind of tell you what is going on when you see pictures like this and what we're actually doing to achieve results that are getting this type of attention. So the first part is just the basic outline of some of the concepts in the study of behavior. And so I want to start out by sharing a little history with you. So the title of the talk is actually something I stole from this book, which is very famous to scientists who study the neural basis of behavior, written by Nicholas Tinbergen in, I think, the 1950s. And so Tinbergen was an ethologist, someone who studied animal behavior in the middle of the 20th century. And he's really famous for being one of the first people to try and systematically watch and come up with scientific explanations for behavior and the way it's organized and patterned. as well as some predictions for what the neural architecture might look like that actually control those behaviors. Even though at the time that he was doing a lot of this work, the tools just weren't available to actually tease apart what the neural basis was for the things he was observing. Nonetheless, he came up with some very simple definitions and some really nice conceptual insights that help us think about things. The first is just to define behavior itself, which is very simple and intuitive. It's just the total movements made by an animal. So animal is moving around in its environment, and everything that it's doing while it moves is its behavior. And ultimately, we want to understand the neural basis of that behavior. What's going on inside the animal's head that allows it to actually perform the actions that it does. And innate or instinctive behaviors are those that are not changed through learning, or those that require no training for the animal to fully execute them. And there are many of these you can think of, and we'll touch on some of the things that animals can do without prior training. But another distinction he made that's very important and interesting is between external and internal stimuli that modulate the animal's behavior. Right? So the first category, external stimuli, are the things we're the most familiar with, basically. Things in the outside world that enter into our nervous system through our normal sensory channels, our eyes, our nose, our tongue, our sense of touch, and they modulate our behavior, right? We perceive things that are happening in the outside world, and that affects how we're behaving, how animals are behaving. But we can also talk about internal stimuli, and these we can think of as our basic drives or motivations, such as hunger or thirst. So basic sort of internal states that the brain can generate that compel an animal to behave one way or the other. And we also think about internal stimuli in the form of signals that originate within the body as opposed to outside of the body. So this can include things like hormones or nutrients that circulate through the bloodstream and ultimately get to the brain, where they're integrated together with information carried by other neural circuits about things in the external world. And it's really the integration of the information that starts on the outside together with information that originates on the inside about what's going on in your body that helps the brain make decisions about how to regulate an animal's ongoing behavioral choices. And so historically, you can sort of think of two basic approaches to the study of behavior. The first is related to what I was just sharing with you, and that's what I'll call the ethological approach. And so this just involves going out into the field or in the laboratory and watching fully intact behaving animals, ideally in their natural environment, right? And this is really useful because it's really what we want to understand. We want to understand the full organism moving around in its natural environment ultimately. But the disadvantages of this approach are that it's subjective, right? It involves human beings literally sitting down and watching animals and taking notes. And our sensory perception isn't quite fine enough to detect all the subtleties and details of animals' behavior while it's happening. And it lacks mechanistic insight, right? You can't go out into the field and watch an animal behave and know what's going on in its brain that actually allows it to perform these actions. This is another famous ethologist from the early 20th century, Konrad Lorenz. Some of you may have learned about him in high school, biology or something like that, because he was the guy who figured out, among a lot of other things, that if you are the first thing that a baby goose sees when it hatches, it'll imprint on you and follow you around whenever you're walking around. So him and Tinbergen were ethologists, and they really watched animals in their natural environment. But then running in parallel to this was a separate field, and I'll call this general approach to the study of behavior the neurophysiological approach. And the advantages of this approach are that you can conduct very controlled experiments that allow you to study the activity of neurons in a very rigorous and quantitative fashion, right? So, this might involve culturing neurons in a dish or taking a slice of brain tissue that you can then stick a metal electrode into individual neurons and study very precisely and with quantitative precision exactly how those neurons are generating the firing patterns that they display. But the disadvantages are that these experiments are highly constrained, right? They're done in very reduced, non-naturalistic conditions. They're completely divorced from the context in which the nervous system actually evolved to function, which is inside a fully intact animal controlling its behavior in the real world. And so as we go through the talk, what I hope you'll see is that some of the tools that have been developed recently are allowing us to basically bridge this gap so that we can actually watch and study animals that are fully intact and moving around, but with tools that allow us to make rigorous and quantitative statements about what's actually happening inside of the brain as that's happening. Before we get there, I want to talk about some of the basic concepts in the study of of behavior, and especially motivated behavior. And if you've ever taken a high school or even an introductory college biology course, you'll often hear this summarized as the 4 F's of evolution when you're starting to learn about what some of the basic motivations and drives are that organisms display. And so the first F is fighting, right? Most animals have some sort of behaviors that they display in certain contexts, either offensive or defensive. And these are typically related to defending a territory or in competing for mates. The second F that's often referred to as fleeing, or the instinct or drive animals display to avoid threats such as predators, right? And this is obviously a survival advantage. The third is feeding, right? And obviously the body needs certain caloric and nutritional demands to be met for the organism to be able to do all the things that it actually evolved to do. And the fifth and most fundamental F is reproductive or mating behavior. And all of these things are highly interrelated, right? So ultimately an animal is going to want to, from an evolutionary perspective, survive long enough by avoiding threats, defending its territory, getting food and other things so that it can one day reproduce. But of course, these aren't the only motivational drives that govern an animal's behavior. There are actually many. So when we say motivational drive, we're talking about states that an animal's brain can put it in that compel it to achieve a specific goal or set of goals at the expense of others, right? And these include things we're all familiar with: hunger, thirst, mating, the drive to sleep or thermoregulate, and many others. And what's important to understand about all these things is they're under tight homeostatic regulation. And homeostasis is a very basic fundamental concept in biology. It just refers to the regulation of biological systems such that internal conditions remain stable. We often talk about something called the homeostatic set point or equilibrium. And you can think of this by analogy to a thermostat in your house, right? Some sort of physiological parameter has to be within a very narrow range in order for the animal to survive. And if it goes too high, it has to become lowered. And if it becomes too low, it has to increase. And this is a pretty intuitive concept if you think about it in terms of something like hunger or thirst, right? If it's been a long time since you've had a drink of water, you get some sensations that compel you to go find water, and then you ingest it, and then that set of sensations goes away. But that set of sensations is what I mean when I say drive. And you develop that drive because your body has moved away from some sort of optimal set point or equilibrium that it's trying to maintain. An importantly related concept to these two is valence, right? So when you haven't had a drink of water in a long time and you see a glass of water, it looks very appetizing to you. And we say that it takes on a positive valence, right? So typically we think of things as having a positive valence when they bring us back to our homeostatic set points, right? You see a glass of water when you're really thirsty, it's very appetitive, and it's associated with pleasure. And things that have a negative valence, we typically think of as those things which move us away from our homeostatic set points, right? And this also is a very intuitive idea. But all these things are interrelated in important ways. And so just to schematize that in simple cartoon form, right, you can imagine some sort of continuum from very hungry and food deprived to full or stuffed when you've eaten very much. And somewhere in the middle is your set point or your equilibrium. And your body is basically trying, by listening to your brain, to keep you at some sort of stable equilibrium, right? So if you go very far in one direction and you're hungry, right, this is typically not thought of as a pleasant state to be in. It's very uncomfortable. And you seek out things in the world that will make that go away, right? So when you see a food item and you're in this state, that food item is now associated with going back towards your homeostatic equilibrium. And this item, we said, to take on a positive valence, right? It's appetizing. You want to go get it. And it's going to be associated with feelings of pleasure. And of course, you can go too far in the other direction and become too full, in which case you might feel unpleasant sensations. And the thing that used to have a positive valence, the very same thing, might now take on a negative valence and be aversive, right? And so these are things we're all familiar with intuitively, but this is sort of the terminology that we often use in behavioral neuroscience to think about these things, right? We have some fundamental drives that are based on a set of homeostatic set points or physiological parameters that our body and brain are trying to keep within a very narrow range. And this affects the value that we attribute to things, both in the external world, in terms of the objects that we perceive, and in our internal world, in terms of the states and sensations and emotions that we feel. And so when we talk about things like motivational drive, one of the key brain regions involved in this is the hypothalamus. And this is a tiny brain region located basically in the middle of the brain. And it contains circuits that are highly conserved, evolutionarily speaking, across all animals. And these circuits control our basic motivational drives, right? So it's located right in the middle of the brain, essentially at the bottom. In the human brain, it's actually less than 1% of the mass of the entire brain. But despite its relatively small size compared to some of the other structures, especially in the human brain, it contains this very important and very highly conserved circuitry, right? And so some of the examples that I'll get to when we're talking about laboratory rodents and things are very interesting and cool, as you'll see. But what I want to emphasize is that the same circuits doing, as far as we know, the exact same things exist in each of us. But if you were to zoom in and look at some of the neurons in the hypothalamus that can control some of these basic motivational drives, they have some properties that make them very useful for doing this. And one is that they're really good at integrating information from both the brain and the body. So what that means is that these neurons are listening to not only other neurons that are carrying sensory information about the external world, but also signals that originate from within the body of the organism. And those internal stimuli are often coming in the form of hormones or other small molecules that circulate through the bloodstream and actually make it all the way to the brain and get to some of these neurons, which have receptors equipped to sample them from the bloodstream, right? So you're taking in information about the outside world through your eyes and your ears and your other sense organs, and that's traveling through certain circuits to the hypothalamus. And in parallel to that, this information from within your body is also getting to these circuits, and that information is integrated by some of these neurons in the hypothalamus. And that integrated information is used to coordinate both the brain and the body in order to produce adaptive behaviors. And a simple way of thinking about how this happens is that these hypothalamic neurons are sending neural signals after they integrate all of this information to coordinate multiple aspects of the brain and the body by sending neural signals to multiple regions in parallel, right? So they're projecting to many different parts of the brain at the same time and sending information to all those places at the same time in parallel. And so when I talk about neural signals, what I mean is that neurons have the ability to send binary signals to one another, basically, in the form of something we call a spike or an action potential. They can send these electrical discharges that happen very fast in time, and that's how they communicate with one another. And they do so by changing the charge that's on the inside of their membrane compared to the charge that's on the outside. So if you actually zoom in on the cellular membrane of neurons, what you'll find is a bunch of ion channels, little basically molecular pores that can open and close to allow charged particles to go into or out of the cell. And when enough of those charged particles go into the inside of the cell, it reaches some critical threshold and it sends a single spike or many spikes, or what we can just call neural signals, to other regions of the brain. And so the important concept here is that these neurons in the hypothalamus are integrating both neural signals in the form of this type of communication coming from elsewhere in the brain and hormonal and other signals coming from within the organism's body. They're integrating that, and they're using that to coordinate multiple downstream circuits that will ultimately drive the animal's behavior. And so in the next part of the talk, I'm actually going to talk about some of the tools that people have developed to study behavior of whole animals, to manipulate and record the activity of individual neurons, and then we'll move into some of the recent discoveries that have come from the development of those tools. But first, I want to break for questions. So if anyone has any questions about the first part. No, it's those are all separate brain regions. So it's sort of in front of the brainstem and the cerebellum, but it's its own region. So I can just kind of go back and point it out. But yeah, so this little dark-colored blob in the center is the hypothalamus. The thalamus would be sitting right on top of it. You've got the cerebral cortex, which is the big wrinkly thing that you always see that's really prominent in human beings. And then you have the cerebellum there, which you mentioned, and the brainstem back here. So the hypothalamus does have a lot of connections to many of those places, including the brainstem. But it's basically sitting sort of on top of or in front of the brainstem. Okay, so part two is going to be a description of some of the experimental techniques that have recently been developed that are very exciting. They're allowing us to basically bridge that gap between the ethological approach and the neurophysiological approach that I mentioned in the beginning. And so the first approach are computational methods that allow us to make unbiased observations of behavior. So this is essentially ethology on steroids. And so first I'm going to show you some movies, and then I'll explain to you what's going on. And so what you're going to see here is basically a heat map of a mouse walking around in an arena. So this is a bird's eye view. You have a square arena that's made out of metal that the animal's freely moving in. And there's a 3D camera above the mouse watching it. So we have information about where the animal is in space, as well as its elevation, how close it is to the floor. And that's what you're seeing here. Right, so the mouse is walking around, and notice that dot that appeared on his body. And so that dot was denoting the onset of a particular behavior. So just sort of notice what happened, right? The animal was sort of turning in one direction. So we can actually do this in many animals and many separate trials and see what that data looks like all together simultaneously, right? So this is many mice. They're not walking around in the same arena at the same time. We've recorded them all once at a time. This is actually data from a former PhD student and friend of mine, Alex Wilchko. And this is the same behavior being elicited by many animals across many different trials. And so just notice what they're doing. The white dot comes on, and they all turn to the right in this case. So it's very simple. It's very obvious what's happening. But what's remarkable is that a human being didn't look at these animals and decide that the animal was turning to the right or decide when it was turning to the right. Actually, sophisticated computer algorithms pulled that out of the data automatically. And then afterwards, human beings came in and gave it an intuitive name. And so the very simplified way that this works is that you have an animal walking around in some sort of enclosure, like an open arena. You have a 3D imaging camera above it, keeping track of where it is. And then you feed that data to a computer. And through the application of machine learning algorithms, you can actually extract from that data information that basically allows you to parse the animal's behavior into discrete units. So you can actually objectively and mathematically define discrete behaviors without any human involvement or subjectivity, right? So there isn't a human being watching and deciding that that behavior is happening and then this other behavior is happening. You're simply feeding the data into a computer and defining mathematically what all of the possible behaviors and poses that this animal takes on are. Then we can go back and give those things names that make sense to us and start to understand how they're sort of put together in different sequences when the animal does different actions. So I'll give you just a few more examples of some of the things that some of these algorithms have pulled out of the data, right? And I just want to emphasize that we didn't know how many behaviors there were going to be, and there are many. These were automatically pulled out, and then after the fact, we sort of looked at them and saw what we were looking at, right? So this is 6 different behaviors, many mice performing each one, and I'll play some of these multiple times. But we do that, and then we give them all, these cute names. But the animal's doing different things for these different ones. So here's one of my favorites. They called it run, forest, run, because as you can see, the dots come on, denoting the onset of the behavior, which is just the animal running kind of swiftly for a couple of seconds. And then you've got a bunch of other ones, right? So this one's called pause, and you can probably predict what's going to happen. The animal just pauses for a brief moment. And you've got a huge variety of others. There are several dozen behaviors, many dozens of behaviors that mice will actually spontaneously display under these conditions. But the takeaway message that I want to emphasize one more time is that there was no subjectivity involved in parsing the data in this way, right? Computer algorithms were developed that automatically figured out all of the different things that the animals were doing, right? So now we sort of have a catalog of all of the different behaviors that the animal does. And we can use some of the other techniques that I'll tell you about in a moment to start to map all of those behaviors onto the underlying neural mechanisms that drive them. So the next thing we need is a way to actually measure neural activity. And there are a variety of ways of doing this. I'm just going to briefly share one of them with you. That technique is called calcium imaging. And so it's an imaging technique that utilizes the fact that every time that neurons send signals to each other, it involves the influx of calcium ions into that neuron. And we take advantage of that in order to build tools that allow us to literally visualize when neurons are sending those signals. And so what we do is basically molecular biologists have figured out that they can staple 2 proteins together that allow us to make neurons literally glow so that we can see them whenever they're active. And so calcium ions turn out to be very, very important for neurons, both for many reasons, including the fact that they're very important for actually sending the signals. And so neurons have evolved a variety of proteins that are very good at sticking to calcium ions once they come into the cell. So one part of this protein is one of those calcium binding proteins. So anytime that calcium gets near this thing, it can stick to it. And it's stapled to this other thing that basically we've stolen from jellyfish. And so this is a fluorescent protein you may have heard of called the green fluorescent protein. And so when you stick these two things together and perform a couple of other tricks, you now have this protein that's been engineered such that once it's put inside of a neuron, every time that neuron fires, which involves calcium coming inside the cell, the calcium will stick to this part of the protein, cause a conformational change in the shape of this part, and then the neuron will literally light up. Right? And so there's a lot of details I'm not sharing with you here, but I just want to give you a visual of what this actually looks like. Right? So what you're going to be seeing here is a movie of the cerebral cortex of a mouse. And the mouse is just sitting in the dark. It happens to be the primary visual cortex. And the movie's sped up a fair amount. But what you're going to see are a couple of hundred neurons that have been engineered such that they express one of those proteins that I just described to you. And you can just see what happens with your own eyes. So there's a microscope basically pointing into a cranial window that's been surgically implanted over a mouse's cerebral cortex. And you can literally watch the neurons as they fire signals from one to the other. And so this technique's powerful for a number of reasons. I mean, obviously, you're measuring when the neurons are active. But the advantage of an optical approach taken through a microscope like this, as opposed to standard electrophysiological approaches, is that you can simultaneously record hundreds of neurons at the same time. Right? And the brain has a heck of a lot of neurons inside of it. And so to really understand what's going on, we really need to record from very, very large numbers of them at the same time in order to figure out sort of what the language the neurons are speaking actually is. Right? So now we've got methods for automatically and objectively parsing the animal's behavior into discrete units. And we've got methods that allow us to actually spy on neurons as they're talking to each other in awake and behaving animals. And the last thing we need, and the one I'll spend the most time on, is a method for actually controlling or manipulating neural activity ourselves. And the technique that we use to achieve this is called optogenetics. And it works like this. So neurons, as I mentioned, have a bunch of these ion channels in their membranes. And they're allowing charged particles to move into or out of the cell. And this is the basis by which they send signals to and from each other. So it turns out that there are certain channels in biology and certain microorganisms that have specifically evolved sensitivity to particular colors of light. These include different species of bacteria and algae, for example. And you can imagine why they might have wanted to do this, right? Algae are photosynthetic microorganisms floating on the top of the water, and they want to know where the sunlight is. So the way that they figured that out in evolutionary terms is they've evolved channels that are sensitive to the wavelengths of light that they care about. Mammals, like us, don't have these light-sensitive channels in our nervous system, right? So the last example of calcium imaging, I showed you how basically molecular biologists figured out, they could take a protein from a jellyfish and take this other protein from neurons and stick them together in a way that allowed us to use them in this as this very useful tool to read out neural activity. And it's a similar story here, where biologists basically stole a protein found in other organisms, put it inside of the neurons in mammalian and other nervous systems, and now we're using that to actually study the brain. So the way this works is you put these light-sensitive ion channels into neurons that would not normally express them. And now, through the delivery of light in very precise ways, you can actually control the firing of these neurons. Because every time you deliver that light, these channels open, they let ions, charged particles, to flow into or out of the cell, and that directly controls the spiking that the neuron displays. Right? So this is very powerful, because now we can take nervous systems inside of awake behaving animals, and we can actually ask, okay, what are the neurons doing while the animal performs a behavior? But also what happens if we turn a neuron off or turn a neuron on at a particular time? We actually figure out not just the correlations between activity and behavior, but actually start to make causal statements because we're actually forcing the system to become on or off at different times. Right? And so this is what you see in a lot of the images that have been in the news lately. Right? You see an organism like a rat, right, that's just been genetically engineered so that some of its neurons, very specific neurons, express this light-sensitive ion channel. And then by the use of surgical procedures to implant very thin fiber optics into the brain, experimenters can actually deliver light at very particular timings to specifically those neurons and control their activity. So basically, in the last part of the talk, after we break for questions, I'm going to share with you some of the results that have come from applying techniques like this, and just kind of show you some of the cool things we've seen. And then I want to end by talking about what some of these results and what the development of some of these tools might mean for human health. So does anyone have any questions before I move on about how the techniques work?
You clearly drilled a hole in the skull of this animal.
That's right.
How do you know that your observational techniques aren't affecting behavior you're trying to observe?
The observational, oh, like the fact that we did all that?
Yeah.
Well, you have to do usually a series of control experiments. So for example, and this will be, I think, even more clear once I get to some of the videos in the next session. But let's say you stick one of these fiber optics into the brain of a rat when it's fully anesthetized under very carefully controlled surgical conditions. And you're controlling neurons and seeing some sort of behavioral effect. And the question is, how do we know that the effect we're seeing is actually real or not just some artifact of the fact that we stuck this thing into an animal's head? Well, there's a couple of controls you do to tease that apart. One is you have the animal that has the whole shebang. It has the neurons that are sensitive to light. It has the fiber optic. You turn the light on and you see what happens. You then have another set of animals that have been injected with a sham molecule that doesn't actually do this. And then you do the same surgery and you turn on the same light. So everything's the same, except the neurons inside the animal's head aren't actually responding to the light, and you see what happens. So any artifacts due to the actual procedure itself will be seen in that second animal in addition. Yeah, that's a great question. It's something that I skipped over. But the way that we get things like this into the animal and things like those calcium indicators that allowed us to actually visualize the neurons as well, there's a couple main ways to do that. The most common is a viral approach. So basically, a lot of viruses have been engineered to contain within them not the normal material they would inject for the purposes of infecting a host, but actually the genes encoding things like this protein. So you take some of these engineered viruses and you inject them into the brain. And the brains of a lot of these animals, like laboratory rats and mice, are very special in that they've been engineered such that even though the virus infects all the neurons it touches, only a very specific subset of neurons in the brain will actually be competent to make this thing that the virus is encoding. So just to reiterate what I just said, you would literally do a surgery in an animal where you inject the virus into its brain. The virus is basically doing the heavy lifting of getting the gene encoding this protein into the DNA of the neuron. So now the neuron starts making it and putting it into its own membrane. And that's basically how it works. The second approach is a transgenic one where you instead of using a virus, you specifically breed genetically engineered mice or rats together in ways that allow the expression of these things based on the animal's own genetics. Okay, so in the last section, I want to talk about some of the recent discoveries that have come from applying these types of technologies to, yep.
So I think now we're actually going to have an intermission.
Oh, intermission, I'm sorry. Okay, cool.
Yeah, so let's just take 10 minutes. If you have any more questions, feel free to go talk to Nick, and then we'll come back to the last section. You're on a roll.
Yeah.
stolen by the end of the series. So if you have your own pen, we really appreciate you just take a few minutes before you leave to fill out this survey and give us some feedback on the topic.
Okay, so the last part of the talk, what I hope is the most interesting part, is basically an application of optogenetics. I'm going to show you applications of optogenetics and some of the behavioral observations that have been made. And then talk a little bit about what the future looks like for this technique. Right? So the first thing I'll share with you is something that I work on myself, which is studying circuits in the hypothalamus that can control feeding behavior. And so here I'm going to show you an example of optogenetically activating or turning on what we can think of as hunger neurons, or neurons that promote feeding behavior. And so I just want to remind you that we're talking about these quote-unquote drive neurons in the hypothalamus that can control specific motivations, in this case, the motivation to seek out and eat food. And that they have this key property where they have both inputs that carry information about the external world coming from other circuits in the brain carrying that sensory information. And they're sensitive to things like circulating hormones and nutrients that are coming into the brain through the bloodstream at very particular locations. And they're integrating both of those things, right? And this makes sense for something like hunger, right? Because we not only learn what food looks like and smells like and feels like, but we also have sensations of hunger that start from within us, right? And literally the gut releases hormones whenever your stomach's empty that actually travel up to the brain and help activate the neurons that I'm going to be describing to you. And we're going to be doing that optogenetically. So this is a technique that we just went over. So basically, we have taken the animal, we have gotten this light-sensitive ion channel into specifically and only these very particular neurons in the hypothalamus. And we now have the ability to implant the animal with a fiber optic cable so that we can deliver light at very precise time intervals to turn on only these neurons, right? And so I'm going to show you a video of what this actually looks like. So the video is sped up by about 50%. And before I let it play, I'll point out a couple things about it. You can see that the mouse is at the top, right? You see this fiber optic cable that's coming out of this attachment at the top of its skull. Also notice that there's this cube of mouse food in the bottom right corner. And basically what you need to know here is that this experiment is done at the beginning of the day, or the beginning of our day. So mice are nocturnal, and they do the vast majority of their feeding at night when it's dark out. And they spend most of their time at the beginning of the light cycle, which is our day, doing nothing. They sleep, and they don't really do much that's interesting. And they certainly don't eat that much. So we do the experiment at that time of day for that very reason. The animal's in a state where it's least likely to eat anything. So now we're going to be turning on these neurons, and we're just going to see what happens. So the simulation's also been on for about 2 minutes at this point. And so by now the animal is sort of perked up a little bit. You can see that he's kind of sniffing and moving and scratching himself. You can see the light flashing on top of his head. And he goes over to that cube of food and begins eating. And they'll actually eat quite a bit. So if you actually quantify it, so under these particular conditions, adult male mice will eat about half a gram of food in 30 minutes. Right? So I'm showing you how much food they eat in this 30-minute period where we're stimulating the neurons compared to how much they eat in 30 minutes immediately after and immediately before. And if you think about it, this is actually a lot of food. So it's about half a gram that they'll eat in 30 minutes. And an adult male mouse of this strain is going to be approximately 25 grams. So it would be comparable to someone of my size eating almost 3 pounds of food in a half hour. So these neurons are really controlling the animal's drive to seek out and eat food. And that is exactly what happens when you stimulate them. But I want to tie this back to another concept we talked about before, which is valence. Because we can also show that neurons like this carry a particular valence. And these particular neurons happen to carry a negative valence. And this actually kind of makes sense intuitively, because if these neurons are controlling the drive to seek out food and they are eliciting feelings within the animal, we think, that might correspond to something we can call hunger, we can think about what hunger feels like to us. And it's not something that we would normally describe as a pleasant feeling. But the way you actually test whether or not a particular set of neurons, when they fire, have a positive, negative, or neutral valence is by doing a test like this. And there are a variety of others. But it's very simple. you put the animal into a two-chambered arena. There's a wall separating each chamber. Each chamber is identical. The mouse can go whenever it wants into one side or the other. And you can use techniques like this such that whenever the animal's on one side, you'll be stimulating the neurons that you're intending to stimulate. In this case, the neurons that promote feeding behavior. And on the other side of the arena, you never do that, right? So as the animal's moving back and forth here, whenever he's on one side, we'll stimulate these neurons. Whenever he's on the other side, we will not stimulate his neurons. And after you've formed that association between the stimulation on one side and lack of stimulation on the other side, you put the animal back into the arena and simply ask, where does he go? Do they like to go to the side with the stimulation, or do they like to avoid the side with the stimulation, or do they not seem to care? So it turns out for the hunger neurons, they actually tend to like the other side. So after you form this association, they will spend most of their time on the opposite end of the arena, and they'll avoid the side that was paired with stimulation. There's no food when that happens, right? Right. So from that, we infer that neurons like this carry a negative valence, right? And this sort of makes sense because it's consistent with what the way we normally think about hunger in our own experience. But it doesn't end there. There's lots of other neurons that have been able to drive a variety of interesting and ridiculous behaviors, including so-called aggression neurons in another region of the hypothalamus. And so this is data that comes from Professor Lin at NYU. And so it's the same technique, optogenetics, and we're shining blue light into the brain to control these neurons. These neurons also have the property of integrating signals from both the brain and the body. They're getting lots of input from centers of the brain that can carry sensory information, in particular from sensory areas related to olfaction. And that's important because mice do a lot of sniffing, right? But they especially do a lot of sniffing of each other. And they use a lot of pheromonal cues to actually identify, for example, the sex of another mouse. And if you put a mouse into a cage with a male mouse that lives there, the first thing he does is he goes and sniffs the mouse's butt. He determines if it's a male or female, and then he will either mate or attack that intruder. But that's the sensory side. These neurons also have receptors that make them sensitive to sex hormones that are circulating through the bloodstream, right? So you can kind of think of them in a way by analogy with the hunger neurons. They're getting inputs both from sensory centers of the brain that are carrying information about the external world, and they're sensitive to factors circulating through the animal's body that originate from within the animal itself. So what you're going to see here is an example of something pretty amazing. So I'm going to start the video, and then I'm going to pause it before we begin stimulating these neurons, right? So initially, we're not going to be stimulating these neurons. So what you see is a mouse in his home cage, and an experimenter has stuck an inflated latex glove in there. It's just a bizarre, novel object the animal's never seen before. So first, let's look at how a mouse normally responds to this. Right? He sniffs it. Mice don't like new things. And he doesn't, he goes to his corner. He'll come check it out again. Now, in a moment, there's going to be a light up here that goes on that's going to signal to us that we have begun stimulating this other group of neurons in the hypothalamus. And then you'll notice that the animal's behavior changes. OK, so now we're stimulating. And the mouse literally starts attacking the glove.
Wow.
Yeah. Yeah. Right. So there's been a variety of neurons that control different motivational drives. I've shown you the feeding neurons and the aggression neurons. There's neurons that control thirst. If you turn them on, an animal will immediately stop what it's doing and go start drinking water. There's neurons that also control mating behavior. They're actually very, very closely related to these aggression neurons. And there's neurons that will actually make an animal fall asleep, for example. So you can take some neurons in the brainstem, actually, and you take an animal during the night period when animals are normally awake, and you turn on these neurons and they'll fall asleep within a few minutes. So basically, optogenetics has allowed scientists to go into the brain and circuit by circuit start cracking exactly what the types of behaviors each individual circuit is controlling. And that's very powerful because we can go from simply making correlational statements about what neurons are doing in the brain to actually making causal statements, right? We can turn on neurons or turn off neurons at inappropriate times and see if we can make or break different behaviors, right? So to conclude, I want to emphasize that the hypothalamus, which is what I've mostly talked about today, is a key brain region controlling basic motivational drives that are common to all animals. New technologies are allowing neuroscientists to rigorously observe behavior, record neural activity, and manipulate neurons in awake behaving animals. And basically the next step for neuroscientists studying behavior is to combine all of these simultaneously so that we're not only observing the animal's behavior in very rigorous and objective ways, but we're actually recording neurons simultaneously so that we can start to correlate what those neurons are doing with what the animal is doing at that time. And we can use techniques such as optogenetics and some others that I didn't mention to actually manipulate neurons during behaviors. And techniques such as this, as I mentioned, allow us to attribute causality to specific brain circuits, right? So we're basically trying to figure out what individual circuits are doing and how they're doing it. And importantly, how they might be going awry under pathological conditions. And so I just want to spend a couple quick slides talking about potential applications of some of this technology to human health in the future, right? So first, I just want to go over what are the basic therapeutic methods we have for treating neuropsychiatric and neurological disorders. Well, first is therapy, right? And that can be highly effective for certain people with certain types of neuropsychiatric disorders. But it has variable effectiveness. It doesn't work for everyone who has a particular disorder that it might work for. And it certainly doesn't work for certain problems, right? It's not something you apply to a neurodegenerative disorder or something like that. We also have pharmaceutical approaches where we give people drugs, but as we all know, these also have variable effectiveness. They commonly have multiple side effects, right? Because it's very, very difficult to make drugs that are very specific, because the brain is this really complicated conglomeration of cell types that have all sorts of different receptors and proteins, and many of them are redundant with one another to one and only one cell type, and it's really, really hard to make a drug that has specific effects that are as specific as one would want. And lastly, there's surgery, right? And this is usually a last resort because it's highly invasive and it's irreversible, right? But it can be a highly effective approach for removing a tumor, obviously, or treating something like, say, intractable epilepsy when all other approaches fail. But it involves basically irreversible damage to a certain part of the brain. And it can also be used for things like intractable forms of depression. So people who don't respond to any other forms of treatment for depression and who have very severe depression will actually undergo something called deep brain stimulation. About afterwards if you want. But literally what that means is that a neurosurgeon goes into the brain, sticks a metal electrode, a very thin metal electrode, deep into the brain, and electrical current is put into a particular part of the brain. And for some reason, somehow, this seems to be a very good way of treating certain forms of depression and other disorders that are not treatable through other means, and no one has any idea how it works. But you can imagine how something that's a little bit more specific, rapid, and reversible might actually be useful to treat things that have been very difficult for us to treat through those traditional methods. And so we can actually start to think about optogenetic forms of therapies. And so a very recent study in mice is something that I'll mention to you to give you one point of reference for how this might look. So something that's really common in human beings, millions of Americans actually, is chronic pain. And it's very debilitating because it's literally pain that has no obvious cause. And a common method for treating it is painkillers, opiates, right? And these have a variety of disadvantages. One, they don't tend to work for forever. And two is that they're highly addictive. But what's happened recently in laboratory mice is that scientists have implanted into these mice, you can kind of see two little dots glowing on each of those mouse's back. What they've done is implant a very small, flexible device that can emit light. And what they've done is they've put into some of the sensory neurons in the peripheral parts of the animal, in the skin, that carry information about pain. And using optogenetics, they can focally apply light to just those neurons and actually control the animal's pain, right? And you can read that out by literally watching how the animal reacts to painful stimuli. And so you can imagine, and this is being pursued by people, developing small implantable devices, for example, and getting some of these light-sensitive proteins into neurons, such as pain neurons in the peripheral parts of your skin, and actually using that to very rapidly and reversibly and wirelessly control those neurons, right? So if someone's suffering from chronic pain, we typically think of that as being caused by some of these so-called pain neurons firing inappropriately. And now we have a method where, conceivably at least, we can just turn them off. And that's exactly like the type of thing you'd want to do. And it wouldn't require treating them with something like an opiate. And you could also imagine it would be useful for other forms of neurological problems that are really tough to treat, such as intractable epilepsy, as I mentioned. So if someone has a severe form of epilepsy and they're suffering from many seizure attacks, the sort of last resort is typically to go in, and neurosurgeons will figure out during an open brain surgery where exactly the problem area of the brain is, and they literally cut it out. And you try to cut out as small an area as possible for obvious reasons, but obviously this is an irreversible procedure, and although it can be highly effective, it can have certain side effects that are unwanted. So you can imagine applying this type of technology to very specifically deliver these light-sensitive ion channels to very specific neurons in just that part of the brain, then using some sort of very small implantable device to reversibly and very quickly deliver light only when it's needed, right? So that's another conceivable application of this. But then, there are other applications you can start to think about that are not science fiction. They're conceivable, but they start to get a little more dicey when you think about them, right? So I mentioned before something like forms of depression that are not easily treated with other methods. And here I'm showing you an X-ray image of what this deep brain stimulation setup looks like. So literally, someone will go in, their depression is severe, and it's not being treated by any other method, and everything's been tried. So the last resort is to literally go in and implant these thin metal electrodes into the brain, and they will deliver at appropriate times electrical current into a particular location in the brain, right? But unlike optogenetics, this electrical current doesn't discriminate against neurons, right? So anything in the path of this is going to get stimulated. And it works for certain things, like certain forms of severe depression and Parkinson's, at least for a while. We have no idea why it works, but you can imagine that if you're going to put someone through an invasive procedure like this, it's conceivable you could also use this type of technology to much more specifically and reversibly achieve something similar. But then, of course, you can start to think about other things that could conceivably be done, and we should probably think about whether or not they should be done, because this isn't science fiction. This is happening in laboratory animals, and people are starting to seriously think about and invest money in applying this type of technology to human beings. And so it's worth thinking about what we might use these for, what we might not want to use these for, and what this type of technology actually means for how our minds work. Thank you. Oh, sorry. I want to acknowledge the different people in Science on the News before we end for questions. Steph and Katie are the co-directors who sort of organize everything, all the events that Science on the News puts on. And then we've got Jordan and Stephen, who are the seminar series directors who coordinate this event that some of you frequent. And then we have a great graphics artist who made a lot of the graphics that you saw today, Olivia Foster. And you can check out the website if you want more information.
Drugs for a while.
Yeah.
They lose their effectiveness.
Yeah.
Similar effect.
I think that's a somewhat complicated answer, but what's nice about this is, and I'm going to speak from what we know from experimental work in laboratory animals. The nice thing about this compared to something like drugs is you can control exactly how long and exactly what the pattern of the stimulus is, right? So we can stimulate neurons at particular frequencies and then stop stimulating them. Whereas a drug, you put it in, it's going to be in your system for hours, and you have sort of no control over it other than the dose at the moment that you take it. Then it has this long period of several hours where it's having very complicated effects on multiple circuits in the brain. But with an approach like this, you can target very specific neurons so that only those neurons at that particular location that have those particular molecular properties are being affected. And the stimulus itself is very temporally precise. So you can deliver the light at particular frequencies at very specific time intervals, and you can shut it off. And so that's a common thing you see in these experiments. For example, in the feeding neurons, where I showed you the neurons that we stimulate that elicit feeding, that stimulation happens for 30 minutes, but it's not a continuous nonstop illumination. It's a very specific stimulation frequency that happens for 1 second at a time and then goes off for 3 seconds at a time. And certainly the neurons will have some adaptation to that signal, but you can actually titrate that to just the level you want it.
If you addict those mice to opiates and then put something in the ear and then stop them from being addicted to opium?
Oh, that's an interesting question. So I can't give you a specific and definitive answer. What I can tell you is that people are using this technology to actually manipulate, turn on or turn off neurons that are very important in the addiction and reward processing stuff that happens in addiction. So there are certain circuits in the brain. There are many circuits in the brain that are probably involved in both reward processing, normal reward processing, and reward processing gone awry, which is basically what addiction is. So the neurotransmitter you often hear about there is dopamine. So there's some circuits in the brain that release dopamine that are very crucial for reward processing and addiction. And people are using this technique to try and turn them on or off or manipulate some of their targets in the brain to see if they can actually relieve or reverse addiction symptoms and things like that. So that's basically the best I can tell you right now. Yep.
The neurotransmitters, you were talking about how there are different neurons.
Mm-hmm.
Do the neurotransmitters affect the neurons characteristics?
Yeah, so basically what I was saying there, so the way we often target these things is based on the molecular characteristics of the neuron. One way that you can distinguish certain neurons from some other neurons is based on the types of things, the types of signaling molecules they release. So in the case of the feeding neurons, for example, they release a couple of signaling molecules that other neurons do not release, and that's what we take advantage of to manipulate just those specific neurons. And so whenever those neurons fire, it seems based on the experiment, the two-chamber experiment I described and some other experiments, that when those neurons fire, the animal basically doesn't like it. And it tries to shut those, it does what it needs to try and shut those neurons off is basically what we think is happening. And there are other neurons that the animals like. So they'll actually go to the side that's associated with stimulating those neurons. And there's a lot of neurons that are related to addiction and reward processing that carry a very strong positive valence. And if you turn them on, animals will work very hard just to get stimulation of those neurons.
When you mentioned the electrical stimulation,
Mhm.
is that the same as electroconvulsive therapy or is that a different technique?
No, this is different. Electroconvulsive therapy is, as far as I know, not used in this country anymore. That's a very, that's
something I remember hearing about when I was young.
Yeah, so that was something that was used a long time ago to treat certain psychological disorders, and it was not appropriate. No, this is literally the implantation, right? This is only for very, very severe problems, such as severe depression that can't be treated any other way, or something like Parkinson's that's neurodegenerative and irreversible. And what they do is they stick a very thin electrode into the brain and stimulate just a relatively small region of the brain, but it's literally injecting electric current. So there's no molecular or cell specificity like you get with optogenetics. Nonetheless, it does have certain very effective treatment outcomes. People have done a little bit of optogenetics in primates. Not much work because it's very difficult. And there's not much more I can say about that. The work that has been done hasn't been nearly sort of as dramatic as making animals eat and fight and stuff. They use it for a lot of experiments that are a little bit more subtle in terms of the questions they're asking. But it's not nearly as widely used as mice and rats. By far the most common organism for studying animal behavior is probably the mouse. Yes. Yes. Yeah, absolutely. So certain of them have been put together already many times. Some of them are just being put together now, and nothing has been done and published yet. So for example, people have used calcium imaging in combination with optogenetics. So you can literally turn off individual neurons and watch what happens to all the other neurons using calcium imaging. So those two have been put together. And then stuff that's just getting underway is combining those observational techniques I mentioned with some of these recording techniques. So people are actually going to record from neurons as an animal does something and correlate that very closely with the very specific behaviors they're recording from that first method that I shared with you. And so that's, I mean, this is really where things are at in the state of the art today. So people are just starting to put a lot of this stuff together. Yep.
Are there any theoretical applications that you're especially excited
In terms of applying something like optogenetics to human health? Yeah, I think yeah, there's a lot you can talk about there. Some of the things are pretty obvious and relatively simple to implement. I mean, it would take many years of development and doing a lot of very controlled studies before it would be doable in humans. But a very simple thing that I'll talk about first is just something like the pain thing. So it's very easy to imagine engineers making small devices that are suitable for humans that can be implanted under the skin and treat something wirelessly by turning on the light to turn off, say, pain neurons, like I mentioned. There are other applications that are very useful, but there's a lot of certain engineering, like molecular engineering, biological engineering problems that have to be circumvented before it's going to be easily achievable in humans. And that involves the details of how we get these things into neurons. And so as I mentioned in one of the earlier Q&A sessions, a common method for doing that is a viral approach. So what we often do when we prepare these animals is we're actually delivering a virus into a particular part of their brain. The virus is then going to basically allow these light-sensitive proteins or other things to be expressed by certain neurons. Now, there are certain advantages and disadvantages of doing that. One is that you readily get a lot of these things into those cells. A disadvantage is it can, with certain proteins, certain viruses and stuff, cause problems if you wait a very long time. So basically one of the engineering problems, bioengineering problems that I think we're going to face before any of this is really going to be doable in the brain of a human is coming up with ways to deliver these proteins to cells in a way that we're completely certain will never compromise the health of the neurons in the brain, even over very long periods of time. And I think there's a lot of interesting challenges there that have to be met before we really start to think about seriously doing some of these things. All right, thank you.